So welcome everyone. We're so glad you could join us today. I am Gabi Conecker. I am president and co-founder of Wishes for Elliott and of the DEE-P Connections project. We're so glad all of you could join us today. We're really excited about this webinar and all of the guests that we've got for our panel today. Thank you for taking time to join us in the midst of all this chaos. I know life is exhausting and busy, and I'm hoping that this is an opportunity for you to think about something else for a little while, and that hoping that you guys are able to take care of yourselves. I know that's kind of a joke, but as best you can. And just a couple of housekeeping things. We're going to, all of the participants are going to be on mute. If you have any comments or questions, please share them in the Q&A or chat boxes that I will be monitoring. If there are any context-related questions, I'll pop in and ask those of the panelists, but otherwise we'll reserve the last half hour or so for questions from participants. And we are recording today's session. It will be available in our resource center in the next 2 days for viewing. I know that will be a lot said today. It's a lot of information, so you'll be able to go back and check out that resource. So without further ado, I'm gonna go ahead and introduce our first panelist. Jenny Burke is mother to Alex, who was diagnosed with an SCN8A related epilepsy. And she's going to share a little bit about her journey with us in terms of genetic testing. She also serves on the board of directors for Families SCN2A. So she takes on a really important leadership role in the DEE community as well. All right, Jenny, so I'm passing it off to you.
All right. Appreciate your having me here today. So my son Alex is 19. I promise I am not going to bore you with 19 years of his day-to-day developmental and diagnostic history, but just a few key things I want to share. He was found to have seizures shortly after birth, spent the first 2 weeks in the NICU. He also had reflux. They weren't really sure what was going on. The epilepsy or the first EEG showed a minor abnormality, but anyway, they sent us home on phenobarbital, and a few weeks later, clean EEG, they weaned him off, and then I don't know, a few weeks after that, bam, he started having all these episodes, and everybody thought they were reflux, except me, who said, this is not normal. I already had an older child, so long story short, they were seizures. He was having generalized tonic-clonics, so back in the hospital. But once we got him on the right medications, he did very well for a couple of years. We did every test in the book that was available to try to figure out what was causing the seizures. Nothing came up positive. There were a lot of inconclusives, things that would have been devastating if he had. He wouldn't have lived very long. So, it was a very difficult time, but because he was developing slowly, but progressively, we decided, we're just going to go with it and do the best we can. Then when he was about 2, seizures started coming back a little bit, and the worse they got, the worse his development got. He started regressing majorly. There were some other comorbidities. He started not wanting to eat. He ended up needing a G-tube. He's also, we found out the hard way, is allergic to milk. But through all this, we just kept going. We kept trying to find the underlying cause, hoping that might give us, a better answer, better treatments. But we had accepted that, we may never know. And we tried to find support in other communities. Epilepsy Foundation was great, but so many people with epilepsy don't have the severe needs that we did. So, and I had some other moms, special needs moms that could, commiserate with, but, we felt pretty much alone. And then when Alex hit puberty, it turns out, he ended up in the hospital for about a month, and just could not get the seizures under control. While we were there, we happened to get the latest round of genetic testing back. We had gone back to our geneticist about 6 months before that, at the very beginning of 2012, and said, you know, we want to keep trying. And she had some more tests. They had gone from having 3 genes on the epilepsy panel when he was born to, at that point, having 53, and now we're up over 100. So it's amazing how quickly science is catching up. And anyway, while he was in the hospital, they happened to hit on SCN2A as being a mutation in his genome. And my husband and I were tested. We did not have it, so it was a de novo variant. Unfortunately, there wasn't a lot known about SCN2A at the time. Our very prominent neurologist said, I don't know anyone studying it. I don't know anyone who has it. We know that possibly sodium channel blockers will be more effective because it's a sodium channel gene. And that turned out maybe to be helpful, but not all of them worked well. Anyway, because there wasn't a lot known, we kind of pushed it to the back of our minds and just went on with trying to take care of our child, who at that point was having seizures every day. And we were just still in that place right now. Actually, he's still having seizures daily. We're still trying to find the right combination of medications. But about 2 years after that, my husband got a funny email from a woman who said, hey, I promise I'm not stalking you, but I saw a video you posted of your son having a seizure, and it said SCN2A on it. And we had posted that because our neurologist was traveling out of the country and he wanted to see a seizure. So Mike posted it on YouTube. And sure enough, that was Leah Schust-Myers, who had started a Facebook group because her son had SCN2A, and he was, I think, 3 or 4 at the time. She said, hey, we found some other families, and we couldn't believe that there were other families out there. So we were obviously so excited. We joined the Facebook group, and one thing led to another, and I'm now on the foundation's board. But in the meantime, you know, because we had this group, researchers became more interested in studying SCN2A. So we went from not knowing anyone and having no research to now having over 600 families around the world and having a lot of research going on. Plus, we found people who there are people. We understand what, you know, each other are going through, and we support each other. And then we find people like Gabi and other disorders that are very similar. And it makes a world of difference. So I'm happy to be able to share our experience, and just encourage everyone, don't give up trying to find, you know, if you don't have a diagnosis or it doesn't seem like the right diagnosis, don't give up, keep trying. You'll find your people and your research team, hopefully. Thanks.
Thank you so much, Jenny. Yeah, it's so incredible to hear about people who've been on this journey for so much longer. You know, my son Elliott is 7, and, you know, it took about 1.5 years to get his diagnosis. And we were told by our geneticist, also, we have no idea about this. We have no idea what this is. We know of 12 people in the world with this.
Right.
And that was, I think it was Japan where it was published. So we're like, either be a further isolation that you find, or it can be a real community. And I eventually, you know, there's so many of these rare diseases, and the communities are exploding. And it's really wonderful to be working with families, SCN2A, and partnering with you all on a lot of things, and all the other patient advocacy groups. So it's really an exciting time.
It is a good time. Yeah, and we were lucky in that our SCN2A had just been added to the gene panel right before he got tested. So
okay, yeah. It's a lot of it's about timing, right?
Yeah, absolutely.
So terrific. Well, I'm going to move on and bring our 2 panelists that are going to be speaking a little bit more about genetic testing and looking at your genetic reports and the importance of genetic testing. First, we have Katie Angione, who is a certified genetic counselor with Children's Colorado Hospital. And she's probably a familiar face to a lot of parents out there because she does the circuit around rare disease groups and does a lot of chats. So we're really grateful that she was willing to work with us on that, this and be with us here today. And then we also have Dee McKnight, who is Medical Affairs Director for Invitae. She's a clinical molecular geneticist, and she's the one who signs genetic reports. She did that for a long time. So you might have her name, her signature on your genetic report. If so anyway, without further ado, I'll pass it off to you all to, I know, Katie, you're going to take over sharing, and I'll be monitoring the chat and the questions. Thanks.
I think you have to give me control to share my screen. Okay. All right, there we go. Okay. So thanks to DEE-P Connections for having us, and to Jenny for sharing your story. It's always really great to hear from families what it looks like on the other side of things. We're going to be going a little bit quickly through these slides, but don't worry, they'll be up to be able to access them after the talk's over, and we're going to try to leave a lot of time for questions. So don't worry too much if we go a little too quickly. I don't have any disclosures. Dr. McKnight is an employee and a shareholder of Invitae. So today we're going to go over a little bit about how our knowledge of genetics and DEE has evolved over time. We'll discuss why genetic testing is important and some of the reasons that, that we bother with doing genetic testing. We'll review some different types of genetic testing, review what a genetic testing report looks like and what sort of the important components are, and then go over how to understand the difference between benign variants, pathogenic variants, uncertain variants, which I'm sure a lot of you have encountered, and then just learn how, what the process is that goes into classifying and reclassifying variants. So genetics in DEE and in epilepsy in general has improved pretty drastically over the past 20-25 years. So you can see when we started out learning about genetics in epilepsy in the 90s, we were learning more about genes that have to do with familial epilepsy. So all these genes in black have to do with familial epilepsy. The genes in red are the DEE genes. So at first, the studies that were being done were mostly family studies that were looking at epilepsy that was being passed from generation to generation. And those were the relatively more mild cases of epilepsy. So it wasn't until about 2001 that the first DEE gene was identified in SCN1A. And since that time, we've kind of had a big gap in identifying those genes, but in about 2010, 2011, things really took off. And with new technology, improved technology and testing methods, we've been able to identify a lot more genes since we first started out. So I think a lot of you have probably experienced kind of what Jenny was just talking about and what Gabi was talking about, is that it is kind of all about timing. When testing was done is going to determine which genes we know about and which genes are actually being tested for. So that's helped us a lot. Diagnostic yield is higher for patients with DEE compared with other types of epilepsy, especially when seizures are starting very early on in life. But we're still not great at detecting and identifying genetic diagnoses in a lot of patients. So these numbers are from 2015, so luckily things have improved a little bit, but the majority of patients still do not have a genetic diagnosis. So testing is always improving. A test that's done today can give you a lot of information than the same test might have done a few years ago. So a couple of examples would be anyone who's had or whose child has had a chromosomal microarray. That test now, compared to about 10 years ago, is going to be able to detect smaller and smaller genetic changes, extra pieces or missing pieces in the DNA. Another example is an epilepsy panel. The panels that we were most commonly sending, even just 5 years ago, were only about 75 genes. Today, the test that we most commonly order for epilepsy is over 150 genes. So things have doubled in a lot of cases. There's also panels that are thousands of genes. So we've come a really long way in doing testing and in being able to identify genetic diagnoses. And that's really, you know, it's a team effort. researchers, genetic testing laboratories, family advocacy groups, all of those people are kind of continuing to push that gene discovery forward. As people get improved access to testing, we're making these more rare diagnoses. And people who are getting diagnoses through more comprehensive, more extensive testing like whole exome sequencing, once those diagnoses are made, that gives the labs an opportunity to recognize that those genes are implicated in epilepsy, and then they can go back and update those panels. And because panels are, kind of more commonly ordered or a little bit more easy to access than something like whole exome sequencing, that means that more patients are getting those diagnoses, more patients are identified, we're able to do research on a larger scale and get a better understanding of kind of the full spectrum of what those conditions can look like. So there's a lot of reasons that genetic testing is important. One of them, and a question that a lot of families will ask pretty early on when they get a diagnosis, is, can this happen again? Is there a chance that if we have siblings or a current pregnancy or future pregnancy to be thinking about, is there a chance that that child could also be affected? Another question a lot of families ask is, when my other children get older and they have kids of their own, is this something that could happen for them? So those are really important questions that sometimes a genetic diagnosis can help to answer. Treatment. People always want to know, is there something that we can do? Is there a better treatment that we can pursue? Is there something that's going to help control the seizures better? Are there medications that are going to make things worse or that we should avoid? And then, especially these days, in the past couple of years, there's been a lot of exciting research going on with things like gene therapy. So having a diagnosis kind of determines, are we able to target that specific gene or that specific mutation with something like gene therapy? We could have a whole separate webinar all about gene therapy, so I'm not going to talk too much about that, but kind of that first step is making that diagnosis. We don't get to something like gene therapy without all the research that goes into it. So identifying patients, doing things like natural history studies that are kind of understanding what the prognosis looks like and what the course of the condition looks like, and then doing clinical trials to get to the more targeted therapies. Prognosis. There's always a lot of different things that influence. It can be helpful to get a diagnosis. It might tell us, will the seizures improve over time? Will they get worse over time? Do we need to be preparing for that? What developmental milestones can we be hoping for? What does this disorder tend to look like in terms of development? Do we need to be worried about or looking out for anything else? Are there issues with the heart or with the kidneys that we need to be monitoring for? And then for some genes, we know that there's what's called a genotype-phenotype correlation, where the specific genetic change, where it is within the gene or what type of genetic variant it is, might determine kind of what things will look like, if it'll be relatively more mild or relatively more severe compared to the majority of patients with that condition. And then I think it's really important to talk about that diagnostic odyssey. Jenny talked a little bit about that. It's a really serious issue for a lot of families. The average time to diagnosis is about 5 years. And that's a long time to have all that uncertainty and to not know, why did this happen? Was it something that happened during pregnancy? We have a lot of families that come to us and they just have this sense of guilt that it was something that they could have done or they could have done differently. And I think getting a diagnosis kind of helps to ease some of that guilt and uncertainty. And then the reason that we're all here today is a genetic diagnosis helps to make connections with other families, with researchers who are working on research with specific genetic conditions. That's what pushes the research and the knowledge forward. And that's what helps families, I think, to connect and to find partners in going through the journey. So Dr. McKnight's going to talk a little bit about some different testing methods.
Hi, thank you. So which test method a provider chooses is often dependent on the clinical presentation of the patient. So rarely now, but still sometimes, a single gene test is sufficient if the clinical diagnosis for the patient is highly specific to a certain gene or variant. But more common, as Katie suggested, that there's the use of panels. And a panel tests many genes at once, and it's great for broader clinical indications like epilepsy or intellectual disability. And then the use of an exome or genome really is very valuable when a patient has already undergone a genetic or a diagnostic odyssey and still doesn't have an answer, or especially for patients that have complex clinical presentations, suggesting they might have multiple diagnoses. So the most common method for sequencing DNA right now is next generation sequencing. And I just briefly wanted to describe how this works. So the DNA is extracted from the patient sample, usually blood. It's fragmented into smaller pieces, and then it's hybridized, which means we use capture probes or baits, and this binds and catches all the DNA that you're interested in sequencing. The captured DNA is then sequenced, and the output is called reads. And these are basically bioinformatic copies of the DNA that you sequenced. And again, next generation sequencing, or NGS, allows for sequencing hundreds, even thousands of genes at once. Previously, different methods were used to detect gains and losses of DNA. So whether it be a copy number variant or CNV, which is the loss or gain of DNA, like sections of DNA, versus an aneuploidy, which is losses or gains of whole chromosomes. This study is called cytogenetics. And early cytogenetics used microscopes. And the cytogeneticist would look for the patterns on the banding patterns on the chromosome, or use a microscope to count the number of chromosomes in a cell. Later, fluorescence was added, and this allowed a cytogeneticist to now look at certain regions of a chromosome. And then more recently, we saw the addition of multiplex targeted assays, and now we could see losses and gains at a gene level. But it is more common today that the next generation sequencing that is used to sequence the DNA is also used to detect losses and gains of DNA. So through sophisticated bioinformatics, these CNVs can now be detected by measuring the amounts of sequencing reads. So by testing hundreds of samples, we can start to see patterns or a baseline of how many reads you often see for any region of the genome. So if the patient has more sequencing reads than you're used to seeing in that region of the genome, it might indicate they have a duplication. Conversely, if they have less sequencing reads than you're used to seeing, it might mean that they have a deletion. And often a different method is used to confirm these NGS observed events to make sure that it's real. And that would typically be qPCR or Ray-CGH, depending on the lab. We're seeing that exome and increasingly genome are very common diagnostic methods for patients with epilepsy. So the exome interrogates 1.5% of the human genome. It's a small sliver of the human genome, but it includes most of the 20,000 known genes and all the coding regions of those genes, so the exons. A genome, and one more thing I want to point out is that the exomes typically are capture-based NGS, just like I described previously, but they tend to have lower coverage than a panel because you're casting such a wide net. There tends to be less data for any particular region compared to a panel. A genome is exactly what it sounds like. You're now sequencing the entire genome. So not only those 20,000 genes, but all the sequence all around those genes and in between the exons in those genes. The one caveat about genome is that we really don't fully understand that other 98.5% of the genome. So in most cases where a genome is being sequenced and the whole genome is actually being analyzed, that's usually done in a research setting. So the future of diagnostics, especially with DEE, is not always predictable, but I do think, I mean, as our understanding of the human genome and its relation to DEE or other diseases increases, we're going to start to see new trends in diagnostics emerge. And so this is just some of the few that are on my radar for epilepsy. Polygenic risk scores or modifier genes. And these are going to be likely useful in better understanding complex disorders or why two patients with the exact same variant might have a different presentation. We're also seeing the emergence of omics. So we're in the age of genomics, which is the study of human DNA, but we're starting to see the emergence of a transcriptome, which is looking at all the messenger RNA. This is the DNA that's being expressed and transcribed in a cell, or a proteome, which is the study of all the proteins in a cell or tissue, and even a metabolome, which is a study of the metabolism of a cell or tissue. And so on the right, I listed just some snapshots of some studies, which are early examples of these different technologies or diagnostics starting to emerge for epilepsy. Now I'm going to turn the presentation back over to Katie, who's going to discuss the anatomy of a genetic report.
So this is just one example of what a test report might look like. So you can see there's this part here that shows the change in the actual DNA. So C here stands for coding. So this is the coding change, the change in the DNA sequence or the DNA code. This 916 here is just the location, sort of the address within the gene that change has occurred. And then this C to T just means that there's one single letter in that code that's being switched out for a different letter. So the DNA code is made up of 4 different letters, A, C, T, and G. And in this case, we have a C that is changing to a T. So that's the mutation or the genetic variant. This part here is showing the change in the protein. So the change in the amino acid, which is sort of a building block of the protein, which is the product that has some sort of function in the brain or in the body. And same thing, the number is just giving you the location within that protein that this change has occurred. And these letters and abbreviations are saying there was one subunit, abbreviated ARG, and then it's been changed because of the change in the DNA, the change in the protein is this cysteine. So it's a change from one subunit to another. This right here is showing you that according to the lab, this particular variant is classified as pathogenic. So what that means is that the lab is confident that this is a pathogenic or disease-causing change within the MECP2 gene. Every report's going to have sort of an interpretation and a summary with some information about that condition. Typically, it'll cite different research articles that talk about that disease, show where those different pieces of information came from. So if you really want to do a deep dive, you would be able to go and reference those papers that are cited in the report. This is just a report with a different gene, a different lab, but you can see that all of those key components are still there. Every lab sort of has their own style for how they report out genetic changes, but you're always going to be able to find that coding change and the protein change within the gene. So the reason that any of this is important is because in some genes, there is that genotype-phenotype correlation where the specific mutation can actually tell you something about what the course of the disease might look like. And then because it might be important for research or for clinical trials, if they're available now or in the future, sometimes knowing that exact mutation helps researchers and physicians to determine whether your child would qualify for that particular study. Unfortunately, not every test report is so straightforward. So this is what things usually look like, or more often than not look like. Maybe not quite so many variants. I had to show you a very dramatic example. But I think a lot of people that are tuning in right now have probably had some experience with variants of uncertain significance. And it can be really frustrating for us as providers. I know it's frustrating for families because there's already so much uncertainty that you guys deal with, and this just adds another layer of that uncertainty. So there is a lot that goes into trying to understand what different genetic variants mean and trying to interpret them so that we don't have so many uncertain variants. So I'm going to turn it back over to Dr. McKnight to talk about some of those methods that labs use to interpret those variants.
Thanks. So diagnostic laboratories in general follow guidelines for variant interpretation from the American College of Medical Genetics and Genomics. So in 2015, the college published guidelines, and the attempt was to standardize the terminology being used between different diagnostic laboratories, and also to create some guidelines so that would be applicable to a wide variety of tests and diseases. So the standardized terminology that was agreed upon by the college was to use the terms pathogenic and likely pathogenic to describe variants that we expect cause disease, a variant of uncertain significance, or VUS, when we just don't know, likely benign or benign variant for variants that are likely to not cause disease. And in the guidance, they recommended that we use different pieces of evidence to weigh and make these classifications. Some of the evidence that we're asked to consider are population data. So is the variant present or absent in large controlled databases? And then also the type of variant. Is it a missense variant, a splice site variant, a nonsense variant? And what type of variant in that gene is known to cause disease? The clinical findings of the individual with the variant is also very important. And are there molecular in vitro or in vivo studies in the literature for that variant, whether it be an enzyme test or cell staining or a knockout mouse? And lastly, are in silico predictors, which try to determine if the variant is conserved in evolution or among similar proteins. There's also a lot of data sharing now between the different diagnostic laboratories through the ClinVar effort. And this is a public database where participating laboratories are encouraged to upload all their variants and any information about the variant that they can share so that the labs can share data and the data isn't housed in some private database that no one else has access to. And this site is open for all laboratory personnel, providers, individuals, anyone has access to this information. But in many instances, with all this evidence, it's still not enough to definitively distinguish if a variant is pathogenic or benign. And this is where it's really important that the provider work with the laboratory to gather as much evidence as possible to help classify a variant. And some of this evidence that a provider can help with are testing parents for phase. Is the variant de novo, or was it spontaneous in the child? And does that make sense for the gene? It's autosomal dominant. Versus did the patient inherit one pathogenic copy from each parent for a recessive disorder? Segregation is also very important through familial testing. So are there other affected family members with the same variant? And it may be important to do a detailed clinical evaluation. So does the clinical findings of the patient fit the gene and variant that was observed, or would further phenotyping of the patient uncover more features, making this gene variant fit a good match? Lastly, it may be important for there to be additional laboratory tests, whether this be an MRI or maybe a metabolic analysis to again see if that finding's a good fit. But it is important that this is a collaborative process between the provider and the lab, and that the provider really is essential in helping to interpret variants and unexpected results. So there was just a couple things I wanted to point out that are sort of recent advances in genetics that isn't maybe as obvious as some of the other things that we've discussed. One major advance is our access to large public databases of genomic data on seemingly control individuals. And in 2010, we had our first publicly available site, 1,000 genomes. It's exactly what it sounds like. It was 1,000 genomes that was available for geneticists to look to see if the variant they're observing in the patient is in any of these individuals in that database. In 2014, the Broad Institute released their exact database, and now we had access to 60,000 exomes. And then a few years later, the Broad Institute released the NOMAD database, and now we had 70,000 whole genomes. And the reason why I bring this up is that this means we now have access to more data. And variants that once seemed rare or even familial are now being observed in these seemingly unaffected adults in these control populations and may be considered benign. So this over this 10-year evolution of public data, we've seen many updated variant classifications, and this means that we've subsequently had to issue many updated reports. And you already saw this image, but it's a great image by Ingo Helbig. And Katie already made the point that basically the gene discovery has been phenomenal. As the cost of sequencing continues to drop and we're seeing the uptake of more exomes and genomes, we're starting to learn more and more genes that are associated with disease. And so just to reiterate the point that what seems like a comprehensive test maybe 5 or 10 years ago would not be a comprehensive test anymore. Finally, I just wanted to mention the Invitae Behind the Seizure sponsored testing program. This is a no-charge comprehensive epilepsy panel, which includes up to 187 genes. Previously, it had been pretty challenging for some patients to obtain genetic testing. And this sponsored testing program has been really successful in increasing access to genetic testing and subsequently reducing the time it's taken for patients to get a genetic diagnosis for epilepsy.
So in summary, genetic testing is always evolving, always improving. We're learning more literally every day. So things are moving pretty quickly, and it's worth kind of taking another look. If it's been a while since your child's had testing, if they haven't reached a diagnosis, or if they have an uncertain diagnosis, it's worth kind of taking another shot and checking in with your genetics providers or neurology providers and seeing, you know, what do things look like now compared to when that testing was first done. There's a lot of value in repeating and reanalyzing and any inconclusive results, and just in revisiting, even if you do have kind of a confirmed genetic diagnosis, it's worth revisiting that over time, because as we learn more, there might be more robust studies out there. There might be more information about specific treatments or specific ideas about prognosis. The bottom line is that we're always learning more. So make sure that you kind of keep an open dialogue with your providers about genetic testing and genetic diagnosis. Had to throw a picture of my dog up there. So we're going to turn it over to questions. So go ahead and submit your questions, and we'll do our best to answer as many as we can. Thanks for joining us.
Thank you so much, Katie and Dee. That was fantastic. A ton of information. I'm definitely going to be going back and kind of watching this again to make sure I understand everything. But want to go ahead and move us. Let's see. All right, so we've got questions streaming in. So if your report says de novo, I know that it means that it's not inherited, but there's a small possibility it can be still be inherited. So what's the likelihood for inheritance in a de novo mutation?
So the word de novo does basically mean that it's not a variant that is present in mom or dad, but that means that it's not present in the sample that was tested. It's not present in the blood or the saliva or whatever specimen was used to do that testing. So you're exactly right. There is still a small possibility that two parents who have a child with a genetic diagnosis might have another child with that same condition. And that's based on something called germline mosaicism. So that's where there might be a mutation that is not present throughout the parent's body, but might be present in a population of the egg cells or in the cells that give rise to the sperm. So that means that there is a chance of passing down a mutation that's not detectable by testing in a parent. It does vary from gene to gene how high that risk is. In general, we quote about a 1% or a 1 to 2% chance of that happening, but we do know that in some genetic conditions, it is a little bit higher. So one of the examples within DEE is the SCN1A gene. We actually know that there is a little bit higher chance of germline mosaicism in SCN1A. So I think for some of the more newly described DEE genes, it is hard to answer that question because I think we just don't know the precise answer just now. We know that there is a chance, but we don't know those exact numbers for every gene.
So someone as a follow-up to that is asking you if you could talk a little bit more about mosaicism in 1A or others in terms of like what that looks like. About 1A, but I don't know if there's generalizations as well.
Within so for germline mosaicism?
It just says about mosaicism. I'm assuming that's what it is, yeah.
Okay. So we know that it's about a 10% or up to a 10% chance that there would be germline mosaicism within SCN1A. We don't know exactly the ins and outs of why that is. We know that there's certain genes that for whatever reason, there is a higher de novo mutation risk, and that's usually in the father or in the sperm. We know that fathers are a little bit older might have a higher risk of new genetic mutations arising. We know that for certain genes, there's a little bit higher risk of new mutations arising, but we don't know exactly what that mechanism is and why it's more common in some genes than in others to have those new mutations. I think we still have a lot left to learn about that.
Yeah, certainly. Okay, so this is a great one to clarify. How does a parent go about getting a copy of their child's genetic testing report?
So it can be really tricky if it's testing that's been done a while ago, but I would say the first step that I would recommend taking would be to get in touch with the physician that ordered the testing or the genetic counselor that you worked with in either ordering or discussing the test results. That's definitely the best place to start. If you have transitioned to a new provider or a new hospital or care center since that testing was done, usually if you have a genetic counselor there, or if you have just a really great physician that you work with, they can usually help you to identify the right people to get in touch with. So most test reports, you won't have the report, but if you know where the lab, which lab did the testing, if you just go to that lab's website and give them a call and say, hey, I know my child had testing done there, I'm trying to track down the report, they can help you to identify what needs to be done to get that report. It's your child. You do have the absolute right to get access to that test report. So they should be able to send you something you can just sign and be able to get a copy.
That's great. So we're having a couple of questions about whether it's worth revisiting. You know, a couple of people are asking, you know, like, oh, we had sequencing done and there was a diagnosis of one thing. Could there be more going on, and is it worth revisiting that? Especially, I think, for medically complex kids and kids who have a lot of comorbidities, there's always the question, is there more going on than just this mutation?
Absolutely. I see the person who asked the question said that they did their testing in 2018. Mm-hmm. 2 years is worth taking another look. Labs usually recommend waiting between 1 and 2 years. I try to stay closer to that 2-year mark because the bottom line is the more time that has gone by, the more new genes we've discovered in that time period. Depending on the lab that the testing was done, a lot of labs offer a no-charge first-time reanalysis, so it should be something relatively easy to do. I think it's definitely worth doing it. There's a lot more knowledge to be gained, whether it's getting more information about a variant that might have been found the first time around, but there was not a lot known about it, or to even identify new variants that weren't detectable or that were in genes that we hadn't yet discovered the first time that the testing was done. So I definitely think it's worthwhile.
Yeah, and I think this leads into, there was another question about that there was testing done in 2016 and there was a VUS, a variant of uncertain significance. And so it definitely sounds like it would be a good idea to do, so would it be reanalysis or would it be retesting? Different kind of,
So
what's
So if it's a variant of uncertain significance, if there's clinical suspicion that it seems like it might be the answer, my recommendation would be to either reach out to the lab or have the ordering provider reach out to the lab and say, hey, can you kind of take another look at this variant? And they can use all of those methods that Dr. McKnight talked about to see what the interpretation of that variant would be at this point. So that doesn't usually require a new test or a new sample. It's just taking another look at that variant in light of what we know today. If that doesn't give us a clear answer, then maybe it is worth considering more comprehensive or more extensive testing.
That's helpful. And excuse me, so to do reanalysis, typically you call the lab and say, hey, would you mind looking at this again as a parent?
I would always start with the ordering provider as a parent if you're able to. It's usually a lot easier for us to navigate that process. We're familiar with it. We can get you in touch with the right people.
Right.
But I would say if a parent has trouble tracking down the ordering provider or it's not someone that they're working with anymore, I think it's always worth reaching out to the lab directly and seeing how to go about doing that.
Yeah, terrific. So we've got a question here about siblings who are typically developing and they age into adulthood and think about having children of their own. Are there recommendations about if and how they can access testing to see if they're at risk or at significantly higher risk of passing on?
That's a great question. So there's kind of a few ways you could go about doing that. Ideally, if you have the affected child's report in hand, that would be really helpful for their adult siblings to share with their own providers. So whether they are, if they have an OBGYN that they're already working with, that would be a good person to start with. They can help with coordinating genetic testing, and in some cases actually referring to a prenatal genetic counselor who can talk through, they can coordinate the testing, they can talk through recurrence risks if they're present, they can talk about options for testing a pregnancy for genetic change. So that would be a good place to start, would be with the siblings ordering providers, just kind of telling them, hey, you know, I have this family history. We know that there's a genetic mutation. How do I go about being tested for this? There also are some labs, including Invitae, that offer sort of patient-directed testing. And Dr. McKnight may be able to talk a little bit better about that, but that might also be an option, is to sort of go directly to the lab and then they can help put you in touch with the right type of provider to help making sure the right testing is being ordered.
I don't know if you want to jump in, you're muted if you wanted to.
Oh, sure.
Sorry.
Yeah, I mean, I agree. I think that for a sibling, I think it's always important to start with the parents. And if you have a known variant that was de novo in the affected child, the chances that the sibling has it are very, very low. But through doing targeted testing, it is definitely possible to go ahead and make sure that the sibling does not carry the variant. And lots of laboratories will do the direct testing. Some of them will do it at no cost. Some of them will be a cost. It's usually not as expensive because it doesn't involve such large methods. We're just going in and looking at that one variant.
And I think it's important to point out that you know, it's going to be different for different genes. I think SCN1A it makes a lot of sense to ask that question because SCN1A can be so variable between different family members. You know, you can have one child in a family who has Dravet syndrome and has a very severe clinical presentation. You can have someone else in that family that maybe they had some febrile seizures when they were young. And even though it's the same exact genetic change, sometimes it can look very different in different people. So it's important to kind of be aware of that variation because sometimes it can go undetected in people that don't have symptoms.
On a similar note, maybe you can talk about in terms of like family planning around, what that looks like if you have a child who has, a genetic change and you're thinking about having more children, what steps you might take.
I would definitely recommend talking to your child's provider and seeing if they can refer you to a prenatal genetic counselor. I can speak a little bit to some of the options that are out there, but they're going to be the experts on the current technology, the current testing methodologies. So again, we would start with doing parental testing if it hasn't been done already. If families are still concerned about that risk for germline mosaicism, there are tests that can be done either very early on in a ongoing pregnancy. So there's amniocentesis, a lot of people might be familiar with, is a way that we can test for genetic changes in an ongoing pregnancy. CVS, or chorionic villus sampling, is another testing methodology that's done on the placental tissue and can be done a little bit earlier on than amniocentesis. And then there's also what's called NIPT or NIPS, non-invasive prenatal screening. So for certain conditions and certain mutations, they can actually test the fetus for a mutation by testing a sample of the mother's blood. So we've kind of come a long way on prenatal diagnosis. There's also things like in vitro fertilization for couples who, for a lot of people doing genetic testing on an ongoing pregnancy, it's an option, but what do you do with that information? And often terminating that pregnancy, making different decisions about that pregnancy, that's not an option for a lot of families. So a lot of people will do something like in vitro fertilization, where they can actually test and detect mutations within an embryo before they implant it. And that way they would only implant the embryos that do not have that mutation. That's not an easy process. It's not a cheap process. So there's a lot that goes into that. We have the ability to do all of these amazing things, but sometimes there's a lot of barriers to them. So I think the best person to kind of talk through all those options would be a prenatal genetic counselor.
Yeah. And it's, we went through that ourselves, and it's, there's risk involved with all of those, and that's scary too.
Exactly.
And we have a comment that for de novo mutations, IVF cannot be done to detect mutations, only for parent carriers. Is that true? That was somebody who was sharing that they thought.
I'm just rereading the question.
Yeah.
So I'm not a prenatal counselor, so I don't know exactly what all the recommendations are at this point. I would say for de novo mutations, the chance of that child or that fetus embryo having that mutation is relatively low. So I think there's probably a lot of providers that would not recommend or would not offer that as an option because there are risks and there are a lot of things involved with IVF. So I think that I'm hoping that's the question you're asking. But I would say for mutations that are either recessive, where families, parents that are both carriers of that mutation would have a 25% chance of having an affected child, or conditions that are dominant and might be variable from one family member to another, where there's a 50% chance of passing down that mutation, obviously those are much different risks than a 1 or 2% risk. So I think that's something that providers and people who are doing IVF need to balance when they're counseling patients on whether that's a good option.
Great, thanks. So we have another question about what to do when panels come back negative and what the next steps are, particularly with Cytogenetics, and there's someone who's asking about, these chromosome epilepsy syndromes. So, and they were concerned about a decrease in diagnosis because of these advanced techniques. So I think you see the question there and probably get the gist, but yeah.
Yeah, it's a very real concern that there are sort of steps in that diagnostic process that are starting to be skipped in many cases because we do have these new advanced technologies. And I think that's a really wonderful question. I think a big part of it is just provider education, making sure that we're, we do this constantly with new residents and new fellows that are going through their training and learning about genetic testing as part of that training, we talk about, you know, these are all the tests that you have available to you. This is what they can detect, but this is what they can't detect. And making sure that we're making those providers very aware of the things that could be missed. And the karyotype is a big one. I know that at our center, when we do a chromosomal microarray, we almost always do it paired with a karyotype to make sure that we're not missing those things. But it's not going to be the case everywhere. So I think that sometimes families need to be good advocates, and patient advocacy groups need to be good advocates for their patients in helping to sort of put that education back on the provider. Because the reality is that not every epilepsy center, not every neurology practice is going to have the people that are educated and aware of genetics and genetic testing methodologies. I wish that there was a more universal way to make sure that all providers have that knowledge. I think it is improving as genetic testing becomes a really important part of all types of different, you know, medicine practices.
Yeah.
I don't know. I don't have a good, perfect answer to that question, but
Yeah.
we're doing our best.
Thanks. Okay, we're going to squeeze in one more question, which is asking about why there might be a link between vaccines and seizures, in particular first seizure. This person is talking specifically about Dravet, because that's not the case for all, but there might be some for, you know, for some disorders that there's a clear link. Did it cut out?
Yeah.
So it's a question about the link between vaccines and the onset of seizures.
I'm not a physician, so that's not really my area. I do know that, you know, just in my experience working in a neurology clinic, we do have a lot of patients that ask this question. Sometimes there are seizures that can be sort of, there is an underlying predisposition to seizures, and it's just a matter of what is that first trigger. It might be the first time a kid gets really sick, gets a bad cold. It might be something like a vaccine that might spike a small fever or something like that. So sometimes there can be triggers that aren't necessarily the cause of seizures, and in most cases are not the cause, but they're sort of bringing them out. I don't know if that's a great answer, but
Yeah,
I'm not a doctor, don't get mad at me.
yeah, I'm sure, like, it's very syndrome specific too, in terms of what all of that looks like.
Yeah.
Want to add anything to that? I saw you nodding your head, just mostly in agreement.
Me?
Yeah.
Yeah, I think that Katie answered it exactly how I would, also not as a physician, but I do think it's, you know, likely the little the immune response or the fever that was triggered by the vaccine that set off the seizures. That would have been my guess.
Yeah. Terrific. Well, we are at time, so I just want to thank everybody for joining us. This has really been an exciting opportunity for me also to learn a lot more about genetics, and I still have lots of questions, as I think we all probably do, but you know, we'll continue to learn together, and you know, things are changing so rapidly. So maybe we'll have you all back at a later date to help us think through how things have changed. So thanks everybody who joined us for this conversation. This webinar was recorded and will be online within 2 days. Just a quick reminder that on May 27th, we have a webinar on improving the quality of rare childhood epilepsy care with Dr. Anu Patel, who is a really active member of many working groups trying to work on this, especially for DEEs. And we have a lot of other webinars that are in the works on developing manageable therapeutic routines at home in the midst of COVID, updates on a vaccine for coronavirus, particularly with a focus on the DEE community, and exactly, you know, that idea about how are vaccines being tested for that community, you know, what considerations are being taken. and then how to promote really positive sibling DEE relationships. And we're really excited about all of these and working towards these. So take care, everybody. Stay safe. And thanks to everyone again. Bye, everybody.
Bye. Thank you.
Bye. Thanks so much.